Hello students, welcome to BioWise with Aanchal. Today we are going to study the chapter Absorption by Roots, the process involved. Now this chapter is of good weightage in your board examination. Stay tuned till the last to get the important questions that can be asked from this chapter. Now those who have subscribed my channel, well and good, but if you have not subscribed, please subscribe my channel and then begin with the video. Now in this chapter we are going to study about absorption. Now when you talk about absorption, there are two things involved. What is absorbed and what is absorbing the material. So here we talk about the absorption of water and we talk about the absorption by the roots. Okay, why we need water? Why plant requires water? Now water is required for the process of photosynthesis. Water is required for transpiration. You will be surprised to know that out of 100% of water absorbed by the plant, 99% of the water is transpired. And since it is transpired, the suction force is created by transpiration. We will be studying about this content in this chapter. Then we need water for the process of transportation. And lastly, we need water by the plants for the mechanical stiffness. Okay, mechanical stiffness. Now these are the four needs why water is involved with the plants. Now next point is what is required by the plants for absorption. So that is root. For any root, if you see, you have got, I am drawing the tap root, you have got the main root, you have got the branching done, that is the rootlets, and every rootlet has got thin hair like structure which is called the root hair. Okay. So what is the characteristic feature? What is the characteristic feature of a root to absorb maximum water? So for that we need to rub the board. Let me clear it. Now next, that is the root. Why root absorbs water? What is the characteristic of a root that it gives maximum absorption to the plants? Now root has got rootlets. Root has got rootlets, and those rootlets are further having root hairs. Okay. So this increases the surface area of absorption. That increases the surface area for the process of absorption. Okay. Mostly the root hairs, they have a cell sap, that is the fluid content in the cell. When I draw the diagram, it will be clear. Cell sap is of higher concentration as compared to the soil. Lastly, the root hair has got thin cell wall. The root hair has got thin cell wall. So that facilitates maximum absorption. Okay. So these are the characteristics for the root to absorb water. What are those? Increase in surface area because of the rootlets and the root hair. Then the cell sap in the root hair basically has higher concentration, which helps in the movement of water from the soil to the root. And lastly, the root hair has got very thin cell wall, so the easy diffusion of the nutrients and osmosis process takes place. Okay. These processes will be dealing just now. So let me clear the board. The diagram of the root hair is asked in the examination. Now to draw the root hair, you need to draw the cubes. You know that plant has got a cylindrical shape. This is the root hair. Okay, and this is a root hair which started to grow. Okay, then they have a cell membrane. They have got a cell membrane. Yes, they have got the cell membrane. Now since it is a plant cell, it has got a very large vacuole. It has got the nucleus and again there are vacuoles present. Okay, they have got a large vacuole. This is the nucleus. Okay, so how will you label it? This is the cell wall, the cell membrane, these are the epidermal layer of the root, because it is the outermost layer, the epidermal cells. Okay, then you have got the vacuole. The vacuole contains the cell sap. This cell sap is of higher concentration to allow the water absorption from the soil into the root. This is the root hair. Okay, and you have got the nucleus also. Correct. So this is the diagram to show the root hair growth and development. So here it is still growing, but here it has fully grown. So this diagram is very important. Diagram can be asked for 3 marks, 1 mark for the drawing, and labeling will be of 2 marks. Is that clear? So this is your diagram. Okay, you can take a screenshot. The transportation in plants occurs by 2 methods, that is active transport and passive transport. So we will talk about active transport and passive transport. The first thing is transportation. Now transportation of water, that is called conduction of water in xylem. So it is of 2 types. One is passive transport. Now when you talk about the passive transport, there is no energy involved. There is no energy involved. But when you talk about active transport, here you have the expenditure of energy. Okay, ATP is used. ATP, that is energy, is used. Okay, and here the movement is always from higher concentration to lower concentration. But here you have the movement from lower concentration to higher concentration by expenditure of energy. Okay. So now passive transport, what we will study is imbibition, diffusion, and osmosis, where osmosis is of 2 types, endoosmosis and exoosmosis. Endoosmosis and exoosmosis. Clear? So this is the complete methods that we are going to study in this chapter, that is transportation, where we have passive transport and active transport. Passive transport is of 3 types, that is imbibition, diffusion, and osmosis. Active transport, you have to spend energy, and our movement is always from lower concentration to higher concentration. This is how the minerals are absorbed by the root hair, because it is generally seen that the concentration of ions is more in the cell sap as compared to the concentration of ions in the soil. So movement of ions take place from a lower concentration, that is soil, to the higher concentration, that is cell sap, and this happens by expenditure of energy. So that process is called active transport. But in case of passive transport, the movement of water molecules, passive transport, that is movement of water molecules, that is from a region of its higher concentration, that is you water the soil more, and water is passing from the soil to the root hair from a region of its higher concentration to a region of its lower concentration. The definition of these terms, that is imbibition, diffusion, and osmosis, is given. So make a note of it, children, it will be easier. You can pause the video, take the screenshot. The definition should be written as it is. Okay, the way I have written it, you have to write the definition as it is in the board examination. So this is done. Let me clear the board. Next is some experiments. Now there is a diagram of an experiment given on my side. You can see the diagram. In this diagram you see that there is something pinkish or purplish that is starting to move in the water molecule. Now here what is shown is the process of diffusion. The potassium permanganate basically diffuses in water, and slowly and steadily you will find that the entire color changes to purple. Similarly, there is one more experiment you can see here. This is the experiment to demonstrate osmosis. You can see there is a thistle funnel. In the thistle funnel there is sugar present. There is sugar solution is present, which is considered to be a hypertonic solution. I will discuss about hypertonic and hypotonic in a while, but sugar solution is a concentrated solution, and in the beaker you can see there is water. Now, since there is a highly concentrated sugar solution present in the thistle funnel, which is separated from water by a thin membrane of the cellophane paper, which acts as a semi-permeable membrane, the water molecule starts moving in the thistle funnel and the level of water goes on increasing. Understood? This experiment demonstrates osmosis. You can have any modification to this by using either a cellophane paper. You can also use the bladders of goat, though we cannot get it so easily. Now you tell me, instead of the cellophane paper or any semi-permeable membrane, if we use rubber sheet, then what will happen? Now rubber sheet is a impermeable sheet or a membrane. So water molecules will not be able to move, so there will be no movement of water taking place between the sugar solution and the water. But when you talk about the muslin cloth, it is freely permeable. So the water molecule, sugar molecule will also pass through the muslin cloth in the water, and there will be mixing of solution taking place. So both rubber sheet and muslin cloth cannot be used in the experiment. Rather, cellophane paper or any membrane can be used as a semi-permeable membrane. Now my question is, now you have understood about osmosis, right? You have understood about the experiment to demonstrate osmosis. Now my question is, how long will the osmosis occur? Now osmosis will occur till the concentration of the molecules on both the sides become equal. That is theoretical, right? Till the time the molecule on each side becomes equal, that is the concentration of the salt or water on both the sides become equal, the osmosis will continue. But in reality this does not happen. The same experiment which you have done, we have seen till now, I have used a thistle funnel, right? A thistle funnel was used, and there was a cellophane paper tied to it, and it had a sugar solution, and here you had water, correct? If I apply pressure here, then a time will come when the water molecule will stop moving. The pressure required to stop the process of osmosis is defined as osmotic pressure. Okay, the definition is given in the description. Kindly go through the definitions. All important notes are put in the description along with a self-assessment test. So do the needful. So this osmotic pressure is the minimum amount of pressure required to stop the process of osmosis. In case of plant, the gravity becomes the osmotic pressure that does not allow the water to be taken in, right? In that case, transpiration plays an important role. How? We will come to know. Be tuned till the last if you want to understand the chapter well, okay? Now, based on the concentration of the liquid and the cells present, the liquid can be classified as 3 parts. Okay, so you can have 3 types of tonicity. One is hypotonic, one is isotonic, and the other one is hypertonic. Okay, now hypo, O, that is outside. So water always move outside from the container which contains from the hypotonic solution. That is, if I have a cell, okay, if I have a cell, let us have a plant cell, So what will happen if it is placed in a hypotonic solution? Water will always move out of the hypotonic solution, means water will start entering into the plant cell. So the cell will become fully turgid, okay. The cell will become fully turgid, right. So this is the movement of water that is taking place. Isotonic, if you talk about an isotonic solution, this is isotonic solution, the water moving in is equal to the water moving out. So there is no net movement of water. There is no net movement of water. Understood? Similarly, when you talk about the hypertonic solution, the cell is present, okay. So water will start moving from the cell. Now this is hyper. As comparison to the solution, the cell becomes hypo. So water will start moving out of the cell. So what will happen? The plasma membrane will shrink. And this condition will make the cell flaccid. Okay, this will make the cell flaccid. We do not use the term flaccid. Better term to use is plasmolysed. Okay, it's better to use the term plasmolysed. We don't use the flaccid term for the plant cell. The entire plant can become flaccid, but when it comes to cell, we generally tend to use plasmolysed state or the plasmolysed state or the plasmolysed cell. Is that clear? So here the loss of water will make the cell membrane shrink. You know that cell wall will not change its shape. The cell wall will not change its shape. It is the plasma membrane which will shrink, right. The plasma membrane will shrink inside, and the place which is present between the cell wall and the plasma membrane will be filled with the hypertonic solution, okay, because cell wall is freely permeable. It will allow the entire solution to pass through it. But the cell membrane is semi-permeable. It will allow the loss of water to happen. So the space between the cell wall and the cell membrane will have the hypertonic solution. This question is asked several times in the examination. Even in 2025 board examination, this question was asked to redraw the diagram and and label them correctly. Where is the hypotonic solution? Where is the hypertonic solution? Okay. So this is a very important aspect. You can mark this. Children, if you make a note with me, then it will be easy for you to study for your exams, right? So this is the solution, that is hypotonic, isotonic, hypertonic. Always remember, from the hypotonic, the movement of water is always towards the hypertonic. That is, from a dilute solution, water always moves to a concentrated solution. But when you have to define osmosis is the process of movement of water molecules from the region of its higher concentration. That is, in which solution the concentration of water will be more? In dilute solution. So water moves from a dilute solution to a concentrated solution. Is that clear? Now let us move ahead. So osmosis we have understood, osmotic pressure we have understood, tonicity we have understood. There is another term called imbibition. Now imbibition is generally the adsorption of water by the surface. It is seen by the dead plants, where dead parts of the plant, wood. We see in the rainy season generally the wooden door becomes blocked. That is because of the swelling that has taken place. Even the initial absorption of water by the germinating seeds is by imbibition. So imbibition is a surface phenomenon where water is adsorbed by the surface. Active transport, where energy is spent, as we have already discussed. So certain ions like potassium and nitrate, they cannot be absorbed directly from the soil because of the concentration difference. So the plant has to spend energy to pump those ions inside. Okay, so that is called active transport. Next, what we are going to study about is turgidity and flaccidity. Now turgidity, what is turgid or what is flaccid? Flaccid is when the plant droops down, and turgid is when the plant is mechanically stiff. Yes. So now for that I need to draw a cell. This is the plant cell and this is the cell wall. Yes, this is the cell wall. Now what happens, there is a large vacuole and there is a nucleus, correct? This is a plant cell. Now what happens, this wall continuously applies pressure on the inner content of the cell. Similarly, this is the vacuole and it has got cell sap, right? The cell sap, because of this content of water, there is always a pressure on the cell wall. So, the pressure which the wall puts on the inner content of the cell is called the wall pressure, and the pressure exerted by the inner content of the cell on the wall is called the turgor pressure. Turgor pressure. Now, in case of fully turgid cell, it is fully turgid cell, the turgor pressure is equal to the wall pressure, right? That means turgid cell is actually a balanced state where the turgor pressure equals the wall pressure. By any chance, if the turgor pressure increases, the cell will burst, and by any chance wall pressure increases, the cell will shrink. Is that clear? If the water content in the cells, that is vacuole, decreases, or if the water content of the plant is lost and the plasmolysis has taken place, that time what happens, the turgor pressure is reduced. So, wall pressure increases and the cell reaches the condition of being flaccid, that is being plasmolysed. Understood? Is that clear? So, this is what all about the wall pressure, turgor pressure, and a cell being turgid, a cell being flaccid, and this is the plasmolysed, how the plasmolysed cell is produced. Any doubt? Put it in the comment section, I will explain again. Now the next question is, why is this turgidity important for the cell? Turgidity provides mechanical stiffness to the soft part of the plant, that is why the plant looks fresh. We call the plant looks fresh. Have you ever observed the coriander leaf which is sold in the market? The coriander leaf which is sold in the market looks so fresh because they keep on sprinkling water on it. When you get it at home, keep it in the refrigerator, it droops down. So that is becoming flaccid or limp, and the condition is called flaccidity, and the cells are plasmolysed. Understood? Now this turgidity is also important for the which tries to emerges out of the soil. So that initial push is given because of the turgor pressure. Then turgidity is also seen in touch me not plant, Mimosa pudica. You touch the plant and the plant droops down because the water content from one cell passes into the next cell, making the cell lose water and limp or become flaccid. So the entire plant droops down. So turgidity plays a very important role in the plant physiology and maintaining the movement of water in and across the plant cells. Turgidity is also important to build up the osmotic pressure, or you can say the root pressure. What is root pressure? The initial rise of water takes place in the plant because of the osmosis, and it continues till certain period of time, certain height of the plant. That height is around 2 meters. So that pressure which leads to the absorption of water is called the root pressure, which is dependent on the osmotic pressure. Right. So because of the osmosis, the pressure developed in the root which helps in the pushing of water in the root and the plant is called the root pressure. The definition is given in the description in the notes. Kindly go through the same. Now turgor pressure is also important in opening and closing of stomata. Now opening and closing of stomata we will study in detail in the chapter photosynthesis, where you have potassium ion theory and the sugar concentration theory. For opening and closing of stomata, we will be studying the same in that chapter, that is photosynthesis chapter. Is that clear about importance of turgidity in the plant? Right. Next we talk about the root pressure, as I have already discussed. Now root pressure, you have to remember one thing, there is something called recep X. There is something called recep X. What is recep X? Is the order that you have to remember how the water moves. Now water moves from the root hair to the epidermis, from the epidermis to the cortex cells, from the cortex cells it goes to the endodermis, from the endodermis it goes to the pericycle, from the pericycle it goes to the xylem. This is asked in the examination to arrange in logical sequence. So, remember recep X is xylem. Okay. This is the order in which the water is absorbed by the root hair. Is that clear? Next, before going to the next segment of this chapter, let me clear the board. Next segment of the chapter is certain experimentations. The first experiment is to show that the root absorbs water. In that case, we take a test tube. We have a small plant, right, with the proper root, and it is filled with water with a oil film on it. What does the oil film do? It does not allow the process of evaporation to take place from the surface of the water. Now, here the amount of water getting reduced is directly proportional to the amount of water transpired from the leaves, but here root is shown to you. That means the water is absorbed by the root. So, the level of water goes on decreasing, showing that the water is absorbed by the roots. Next experiment is to show that water is conducted by xylem. Now, in that case, if you just add eosin dye or any dye into the water, it turns pink, and when we go for a transverse section of the stem, we see that it is only the xylem which is being pigmented red, showing that the water is conducted by xylem. Next experiment is to show the phloem, that is translocation of food happens through the phloem. So, if you take a stem and you make a hole here, what will happen? The leaf present here will droop down, showing that the segment that you have removed is basically the xylem which you have removed, and the water is not able to go up, hence the leaf droops down. Similarly, if you take a stem and cut a outer portion of it, this is called the girdling experiment. So, what will happen? The accumulation of food will start taking place, and there you will find a bulge in certain period of time, showing that the food is getting accumulated here, and later on you will find that the plant dies because the root is not getting the food. The root is not getting the food. So, this experiment shows that xylem is for conduction of water, xylem is for conduction of water, and phloem is for the translocation of food, is for the translocation of food. Is that clear? Will you remember this? The last part of the chapter speaks about the forces required for the ascent of sap, forces for ascent of sap. What is ascent of sap? That is movement of water. Okay, that is ascent of sap. Now, what are the different forces required? The first one is capillary action. Now, capillary action, you know that xylem has got tracheids and vessels which forms tiny tube-like structures. So, initial rise of water takes place due to the capillary action. Okay. Then you have the root pressure. Now, root pressure is because of the osmosis that will happen from cell to cell. Now, osmosis will lead to the development of turgidity in the cells, which forces the water from one cell to the other, leading to the movement of water cell by cell in the plant to certain height, that is around 2 meters. So, root pressure is the initial uptake of water that has taken place till a certain height. That is, small shrubs and all, they are happy with the root pressure. It is because the root pressure only we get guttation, that is oozing out of water from the margin of the leaves, which leads to the formation of dewdrops. Okay. Next one, you have the cohesive force of water molecule. That is, cohesive, that is in the same water molecule. And then you have the adhesive force or adhesion. Adhesion is between the attraction between the water molecule and the xylem vessel. So, if you have the xylem and you have the water molecule moving, why my water molecule has got yours H2O, no? So, H2O, bigger one is O, smaller one is, the ears are the hydrogen basically. So, there is a force of attraction between the water molecules, the hydrogen bond. This is the cohesive force, and there is a force of attraction between the xylem and the water molecule, that is the adhesion. Now, these two forces makes the water move in a regular column to the leaves. And the last one, if the plant height goes beyond 2 meters, then it is the transpirational pull. Okay, transpiration is the next chapter. So, we will be going in detail of transpiration too. So, what happens transpiration, like when you take a straw to drink water, we need to suck in the liquid from the straw. That suction force is created by you. Similarly, in the plants, if all the xylem is like tube, there is a suction pressure from the top, which is the transpiration. So, with this we complete the entire chapter on absorption by roots, the process involved. Yes, some of the important questions related to the chapter is displayed here. See to it that you know the answers of all these questions. If you have any doubt, put that query in the comment section, it will be answered. Please like, share and subscribe, and happy learning, children.